Chapter 9, Segment 4. Chapter 9 begins talking about hearing, vestibular perception, taste, and smell. This segment begins talking about the taste bitter. Bitter is associated with many toxic substances such as nicotine and caffeine. Members of the T2R family of the G protein coupled receptors appear to be in bitter receptors. Then there's umami, which is the fifth basic taste, and is described as savory and as a more meaty flavor. The receptor metabotropic glutamate may be responsible for this taste. Taste information is transmitted into several parts of the brain. One important thing to know is the gustatory system extends from the taste receptor cells through the brainstem nuclei and thalamus to the cerebral cortex. So it's just saying how taste travels. Some gustation researchers argue that the brain extrapolates taste information from pattern coding. This type of encoding provides great capacity for distinguishing subtle differences between stimuli, and it provides a rich parallel set of inputs upon which the brain can act and tune to the stimulation it receives. Another section within Chapter 9 is the sense of smell starts with receptors in the nose. In humans, a sheet of cells called the olfactory epithelium lines the dorsal part of the nasal cavity and adjacent regions. We possess three types of cells found in the epithelium: supporting cells, basal cells, and olfactory receptor neurons. If the olfactory epithelium is damaged, it can regenerate and will probably reconnect to the olfactory bulb. Another section, olfactory excite specialized receptor molecules on the olfactory receptor neuron. Interactions of odorants with their receptor trigger the synthesis of second messengers such as cyclic AMP, resulting in depolarization of the olfactory neuron, which leads to an action potential. And then another section in this segment is olfactory axons connect with the olfactory bulb, which sends its output to several different brain regions. The olfactory bulb is organized into many roughly spherical neural circuits called glomeruli. Output for the olfactory bulb consists of the axon from mitral cells, which extends to a variety of brain regions. This close relationship between olfactory inputs and limbic system structures involved in memory and emotion may help explain the potent ability of odors to evoke strong nostalgia of memories, so like smells associated with memories. And then lastly, many vertebrates possess a vomeronasal system. Many vertebrates have a second chemical detection system that appears to specialize in detecting systems that appear to specialize in detecting hormones, the odor signals or trails that many animals secrete. This vomeronasal system is present in most terrestrial animals, and the VNO receptors are remarkably sensitive, detecting extremely low concentration of pheromone molecules. And then trace amine associated receptors. These receptors are expressed in specific olfactory neurons within the main olfactory epithelium, just like the olfactory receptors. But at least in mice, they don't seem to respond to volatile sex-specific urinary compounds that may be pheromones. This concludes Section 4 in Chapter 9. Thank you.